Okay, so welcome back to this next video in which we are discussing asthma and anti-asthmatic drugs. Okay, so we're currently in the process of discussing what happens in an asthmatic attack. So we've discussed that mast cells release these two key mediators, histamine and cysteinyl-leukotrienes. We've discussed that histamine and cysteinyl-leukotrienes cause bronchoconstriction by causing contraction of the bronchial smooth muscle cells, and they also cause an inflammatory response. Okay, now in the immediate phase of the asthmatic attack, you have the bronchoconstriction due to the contraction of bronchial smooth muscle cells, and you also have this inflammatory response occurring, which results in an exudate being formed in the lamina propria of the bronchial wall, okay, and that also worsens the narrowing of the lumen of the airways, basically. Okay, now what we're moving on to is the late phase of the asthmatic attack, in which there is more damage being done to the bronchial wall, basically. Okay, so in the late phase of the asthmatic attack, so long after you've had the initial occlusion of the airways, okay, or reduction in the lumen of the airways, and that's all gone now, okay, so that's remiss spontaneously. Now, I should say that asthmatic attacks usually do remiss spontaneously, okay, so the bronchial smooth muscle cells do relax back down on their own, usually. If they don't remiss spontaneously, then you can go into what's called status asthmaticus, okay? So there is this important term called status asthmaticus, and this means where the bronchoconstriction doesn't remiss, basically, it just maintains there. Again, this is a medical emergency, okay? This is very, very lethal, and it can kill you just simply because you're not getting enough oxygen into your blood, so it can lead to hypoxemia and therefore death. Okay, so status asthmaticus is a medical emergency, and it's just where the bronchoconstriction just doesn't remit, basically, it stays there. Okay, right, but usually in an asthmatic attack, the airway reopens up spontaneously, the airway smooth muscle cells re-relax, and the airway opens back up. Okay, right. Now, in the late phase of an asthmatic attack, which occurs hours after the initial bronchoconstriction due to the contraction of the smooth muscle cells, what's happening is this inflammatory response that was triggered by the histamine and the cysteinyl-leukotrienes is starting to recruit in white blood cells from the blood. Okay, so in the immediate phase of the asthmatic attack, the inflammatory response just contributes an exudate, basically. Okay, so you get edema in the lamina propria, but in the late phase it gets around to starting to bring white blood cells out of the blood and into the lamina propria. And what does it bring in? Well, it brings in more eosinophils, okay, and more T helper 2 cells. Okay, so you make that initial chronic inflammatory response even worse, basically, now, because remember, these are the 2 cells, the eosinophils particularly, which then drive further damage to the bronchial wall and make the airway hyperresponsiveness worse, basically. Remember, they kill airway epithelial cells by their cytotoxic proteins, okay, and they also release the transforming growth factor beta, which causes airway smooth muscle cell hyperplasia and hypertrophy, which increases the mass of the bronchial smooth muscle cell. So this late phase of an asthmatic attack basically makes the airway hyperresponsiveness even worse, basically. Okay, so it's a portion of the asthmatic attack that should not be forgotten. Right then, to summarise what we have now discussed about the pathology of asthma, we've seen that asthma is caused by this chronic allergic adaptive immune response. Okay, now in both non-atopic and atopic asthma, you get a T-cell response, specifically a CD4 positive T-cell response, which generates T helper 2 cells. And we're about to move on to looking at how you get those T helper 2 cells armed against the allergen. Okay, so you get those being produced, and what then happens is they infiltrate into the lamina propria, and they then release those cytokines which recruit eosinophils into the lamina propria. The eosinophils then kill the columnar epithelial cells of the bronchial airway, okay, and also release transforming growth factor beta. Okay, killing the epithelial cells is one of the things that leads to airway hyperresponsiveness, because it now means that you've got holes in your airway epithelium. The transforming growth factor beta then causes airway smooth muscle cell hypertrophy and hyperplasia, okay, which then means that the amount of bronchoconstriction you're capable of producing is now increased, basically. Okay, so bronchoconstriction is now exaggerated when it does occur. Okay, then what can happen in atopic asthma, and this is now just in atopic asthma, is along with all of that, you get a B-cell response against the allergen, which produces IgE molecules, okay? And the IgE molecules then mount on the surface of mast cells on Fc epsilon R1 receptors. Okay, then how do you get an asthmatic attack? Well, in both atopic and non-atopic asthma, you can get non-allergic asthmatic attacks to things like cold air and also noxious gases such as sulfur dioxide and ozone, because when you breathe those in, those can now get to the mast cells because of the holes in the epithelium, and then they can trigger the mast cells to release histamine and cystine-L-leukotrienes. Okay, in atopic asthma, you can also get allergic asthmatic attacks where you breathe in a lot of this allergen that initially causes the problem, okay, and that will now activate the mast cells to release histamine and cystine-L-leukotrienes via the activation of the Fc epsilon R1 receptor, and we saw the exact pathways by which that causes the release of histamine and the production of the cystine-L-leukotrienes. Okay, it's the histamine and the cystine-L-leukotrienes then that actually cause the asthmatic attack. They cause the contraction of the bronchial smooth muscle cells, which causes the major bronchoconstriction. Okay, in addition, they cause this inflammatory response, which in the immediate phase of the asthmatic attack contributes a lot of edema, which further narrows the lumen of the airways. Okay, and in the late phase of the asthmatic attack, this inflammation brings in more of these awful white blood cells which are causing the problem in the first place, so it brings in more T helper 2 cells and more eosinophils, and therefore it makes it more likely that in the future you're going to get more asthmatic attacks because you're now making the airway hyperresponsiveness worse, basically, by recruiting more of these cells in. Okay, right. So that's where we've got to now with the pathogenesis of asthma. What I now want to do is go back and look at the initial allergic immune response. Okay, and I want to firstly look at the one that occurs in both non-atopic asthma and atopic asthma, which is a CD4 positive T cell response. And then I want to look at the one that just occurs in atopic asthma, which is the B cell response. Okay, so firstly then, let's look at the CD4 positive T cell response. Okay, so I want to look at CD4 positive T cell activation in the asthma. Okay, so I want to look at how you can get this allergic adaptive immune response against our allergen. Okay, right, so I should say first of all that we do not understand why some people launch adaptive immune responses against these allergens and then other people do not. Okay, that's not well understood at present. What is understood is that these people who have these allergic adaptive immune responses initiate these adaptive and allergic immune responses. So what I'm going to do is just tell you the story of what must happen, okay, but we don't really know the answer as to why this occurs in allergic people and not in non-allergic people. What is different between these two groups of people? That question is still not very well understood. Okay, so I'm just going to describe to you what must happen, but I'm not really going to be able to explain to you why this happens in these allergic individuals. Okay, so basically it's thought that the cell that initially drives the activation of the CD4 positive T cells is a dendritic cell. Okay, so remembering back to your immunology, the way that you activate CD4 positive T cells is through antigen presenting cells, APCs for short. Okay, so APC stands for antigen presenting cell. Okay, and these are cells which basically process antigen molecules and then present little fragments of them to T cells. Okay, and if they can find a CD4 positive T cell with a T cell receptor against that specific little fragment, then they can activate it. Okay, now one of the major types of antigen presenting cells is a dendritic cell, and these are very numerous in the airways basically, so it's thought that dendritic cells must present the allergen that's being inhaled basically, that drives the asthma. Okay, right, so let me now draw again for you a little stretch of the columnar epithelium of the airways here. Okay, so here are the columnar epithelial cells here. We're going to have cilia on their apical surfaces here. Okay, and then they're all going to be sitting on the basement membrane, which is down here. Okay, now, just underneath the basement membrane then, you have dendritic cells which send their dendrites through the basement membrane and in between the columnar epithelial cells, like so. Okay, so dendritic cells are called dendritic cells because they have these dendritic processes. Now, they're nothing to do with neurons, okay? The dendrites here are very different from the dendrites in neurons. All they have in common is that they're these processes that come off the cell body of the cell. Okay, so dendritic cells are not capable of firing action potentials like neurons. They simply do not have the voltage-gated ion channels that you need in order to fire an action potential. Okay, so I'll colour the dendritic cell here in orange. So these are our dendrites of our dendritic cell. Okay, and for short I'll abbreviate the dendritic cell down to a DC. Okay, right, so basically its cell body is underneath the basement membrane here of the airway epithelium, but its dendrites extend through the basement membrane and in between the columnar epithelial cells. Okay, now, what's then believed to happen is when you breathe in this allergen chronically, okay, you're all the time exposed to a little bit of this allergen basically, maybe because you've got a pet cat or something, okay, and this is this allergenic protein, whatever it might be, okay, what then is believed to occur is that the allergen gets internalized by the dendritic cell. So the dendritic cell is going to internalize the allergen, okay, so if I now draw the dendritic cell a little bit larger here, okay, so let's just draw dendritic cell. I'll draw it a little bit simpler as well. Okay, so here are its dendrites once again. So what it's going to do is it's going to internalize the allergen into a vesicle here. Okay, so here's our allergen within a membrane-bound vesicle inside the cell, and I'll just put the nucleus of the dendritic cell on as well, just to make it look a little bit more cell-like. Okay, and what's then going to happen is it's going to process this allergen. It's going to break it up into loads of little fragments, okay, and it's going to put all of these little fragments on MHC class 2 molecules on its surface. Okay, now I'm only going to show this for one fragment, and we will follow the story of one fragment, but in reality it will have loads of fragments of this allergen on its surface. Okay, so I'll put a little fragment of our allergen here on this MHC class 2, and unfortunately, just because of the crudeness of the picture, the fragment of the allergen looks identical to the allergen, but understand that this is just a little piece of that allergen, a little sequence of the allergen, okay, and you'll have other little chopped up pieces all over on these MHC class 2 molecules. So this molecule that it's bound to is what's called an MHC class 2. Okay, now this does not this time stand for myosin heavy chain. Okay, this stands for the more common meaning of MHC. Okay, which is major, that's the M, and then big word now, histocompatibility, and then it's complex, okay? So the H is for histocompatibility, and then the C is for complex. So don't think that the HC is for histocompatibility, it's not. The H is for histocompatibility, and the C is for complex. Okay, right. Oh, and I've missed out the class 2. So this is the major histocompatibility complex, class 2 here. Okay, right. And I'll just colour in the major histocompatibility complex, class 2 here in blue. Okay, right. So basically, the dendritic cell will process its allergen that it's internalized. It'll break it up into loads of little fragments, and it'll put these fragments on its surface attached to these MHC class 2 molecules. Okay, then what will happen is it's now going to go and try and activate CD4 positive T cells. So it will now drain in lymphatic vessels, of which you will have a few in the lamina propria. So in the lamina propria, you'll have lymphatic vessels which will eventually drain into lymph nodes. Okay, so let me now draw you a picture of a lymph node, because lymph nodes are truly brilliant things. They're absolutely brilliantly designed for what they do. Okay, so lymph nodes basically are these immunosurveillance structures. Okay, they are for the purpose of looking at all of this fluid that is coming from tissues all over the body and trying to spot clues that show that there is an infection, basically. Okay, so what you've got then is all of these incoming lymphatic vessels, all of these afferent lymphatic vessels. These drain into a space around the outside, okay, which is known as the subcapsular sinus. Okay, so this space is called the subcapsular sinus, and I'll just colour in the subcapsular sinus now in green. So this space here in green, which all of these afferent lymphatic vessels are draining into, is called the subcapsular sinus. Now, the fluid that's coming into the subcapsular sinus, and I'll just raise this up a little bit, needs to get into a sinus down here, okay, which I'll colour in red, which is called the medullary sinus. Okay, so this sinus here, this is called the medullary sinus. Okay, and then once it gets into the medullary sinus, it can go into this outflow vessel, this efferent lymphatic vessel. So you've got all of these afferent lymphatic vessels draining into the subcapsular sinus, and somehow the fluid needs to get from the subcapsular sinus into the medullary sinus so that it can go into the efferent lymph vessel. Now, to get from here to here, it has to drain through this solid matter here. Okay, and this consists of absolutely loads of white blood cells, specifically special types of white blood cells which are called lymphocytes, okay, because they dwell in the lymph nodes. Okay, so this solid material here is made up of loads and loads of lymphocytes, T lymphocytes and B lymphocytes, okay, and the T lymphocytes that are here are of the two types, CD4 positive T cells and CD8 positive T cells. So the basic idea is that all of this fluid has to percolate past these lymphocytes, which are looking for any sign of pathogens, basically, okay, and they will respond if they find any sign of pathogens. Okay, so that's how it's brilliantly designed for immunosurveillance. They are looking for any sign of problems coming from this fluid that's coming from all over the body, basically. Okay, or rather, because there's loads of different lymph nodes from many different peripheral tissues. Okay, right, so basically our dendritic cell here, now this has got some allergen fragments presented on MHC class 2, it's going to go in a lymph vessel, and eventually it will get to a lymph node, and basically it's going to percolate through all of these lymphocytes, and it's going to be coming into contact with loads and loads of CD4 positive T cells. Now let me just tell you a little bit more about CD4 positive T cells. Let's draw a CD4 positive T cell here. So I've already told you that CD4 positive T cells, the key thing that they have is this CD4 molecule here. Okay, and that separates them from CD8 positive T cells. The CD4 means that antigen presenting cells presenting on MHC class 2 are capable of activating these T cells. Okay, CD8 positive T cells are instead activated by antigen fragments presented on MHC class 1. Okay, now the other really important thing to say about CD4 positive T cells is that you have absolutely loads of them, and they are all really diverse. So in this lymph node, you'll have absolutely loads of different CD4 positive T cells, and what's different between them is that they all have very different T cell receptors. Okay, so here is our T cell receptor, and the portion of the T cell receptor that differs is the portion that actually binds to and recognizes the antigen fragment, basically, on MHC class 2. Okay, so TCR for short stands for T cell, that's the TC, and then receptor. Okay, so basically, all the different CD4 positive T cells that you'll have in this region will all have different T cell receptors which can bind to different antigen fragments presented on MHC class 2. Okay, so the task now is for this dendritic cell to find a CD4 positive T cell that has a T cell receptor which can bind to one of the many allergen fragments that it has all over its surface. Okay, so it needs to find a T cell that has a T cell receptor which is capable of binding to just one of these allergen fragments. So remember that when we processed the allergen, we chopped it up into loads of little pieces, and we put all of these pieces on MHC class 2 molecules. Okay, we only need a T cell that's capable of binding to one of these allergen fragments. Okay, it doesn't need to bind to more than one of them, it just needs to bind to one of them. So the dendritic cell needs to find a T cell that has a T cell receptor capable of recognizing one of these allergen fragments. So let's assume that it manages to find this. Okay, so let's say here is one of our dendrites of our dendritic cell here. Okay, here is the MHC class 2 on the surface of the dendritic cell here. Okay, so in blue here, this is our MHC class 2, and it's now presenting the allergen fragment on the MHC class 2 to this T cell receptor here. Okay, now I should say something here about the nature of allergy then. So we don't know whether the difference between allergic individuals and non-allergic individuals is that allergic individuals have T cell receptors which have T cells which have T cell receptors which are capable of binding to fragments of that allergen. Okay, that could be the difference between allergic and non-allergic individuals, that allergic individuals just happen to make these CD4 positive T cells with T cell receptors which can bind to the allergen fragment, and non-allergic individuals don't. Okay, alternatively, it could be to do with the dendritic cell. It could be that dendritic cells of people with who are allergic to this allergen present the allergen to T cells, whereas people who aren't allergic don't present it to T cells, and that could be determined by which genes for MHC class 2 you have. Okay, so there are many different components that could determine whether you're going to be allergic to this allergen or not. Okay, right, so coming back to what does happen though in people who are allergic to this allergen. Okay, you present the allergen fragment to this CD4 positive T cell that has a T cell receptor that's perfect match for this allergen fragment, and then what happens is the CD4 then binds to the MHC class 2. So I might just extend that CD4 up a little bit. Okay, it should be binding to the MHC class 2 there. Okay. Right, now this sends the T cell what's known as signal 1. Okay, so this is called signal 1 of activation. Okay, however, to activate a CD4 positive T cell, you need more than signal 1. Okay, you need also signal 2. Now, what provides signal 2? Well, basically, in order for the dendritic cell to deliver signal 2 to the CD4 positive T cell, it also needs to present co-stimulatory molecules on its surface. Okay, so where should I put this? So the dendritic cell needs to have co-stimulatory molecules on its surface. Now, dendritic cells do not put co-stimulatory molecules on their surface unless certain receptors on the dendritic cell have been activated. Okay, and these special receptors on the surface of the dendritic cell that need to be activated in order for co-stimulatory molecules to be put on the surface are called PRRs, for pattern recognition receptors. Okay, so I'm just going to go over onto the other page just to write this down because we're running out of space there. Okay, so PRR then stands for pattern recognition receptor. Okay, so the P is for pattern, the R is, well, the first R is for recognition, and then the second R is for receptor. Now, what do these pattern recognition receptors actually do? Well, basically, pattern recognition receptor is a huge, great umbrella term, okay, that covers a huge number of different receptors that all bind to vastly different ligands. Okay, so what groups them together is that these are all receptors for ligands that you would never ever usually find in humans. Okay, they are receptors for ligands that are associated with bad pathogenic molecules, basically. So bacteria and viruses and fungi and parasites. Okay, basically these pattern recognition receptors are a very broad group of receptors which recognize ligands that would never ever be found on a human cell. Okay, and would only be found on pathogens, basically. And there is an equally broad umbrella term for the molecules that they actually recognize. So this group of molecules that you would only ever find on pathogens are called pathogen-associated molecular patterns. Okay, or PAMPs for short. So the P is for pathogen, the A is for associated, okay, the M is then for molecular, and then the second P is for patterns. Okay, so basically, if the dendritic cell is exposed to some of these pathogen-associated molecular patterns, okay, so molecules that you would only ever find if you had a pathogen nearby you. Okay, so no human cell in its right mind would ever make one of these molecules. Okay, then you can activate these pattern recognition receptors on the surface of the dendritic cell, and these then activate the dendritic cell to put onto its surface co-stimulatory molecules. Okay, and these co-stimulatory molecules are needed to activate CD4 positive T cells. Now, what's the point of all of this? Well, the point of all of this is a checkpoint. Okay, it's a checkpoint to make sure that you are not activating CD4 positive T cells unless this dendritic cell has actually seen some very dangerous PAMPs, basically, some very dangerous pathogen-associated molecular patterns. Okay, so what's the main example of co-stimulatory molecules? Well, the main examples of co-stimulatory molecules are what are known as B7 proteins. Now, there are two different types of B7 proteins. There is B7.1 and there is B7.2. Now, B7.1 has another name. It's also called CD80. And B7.2 also has another name. It's also called CD86. Now, for our purposes, both B7.1 and B7.2 are going to do exactly the same thing, so we will just abbreviate them down to B7 molecules. Okay, right. So if this dendritic cell is going to activate a CD4 positive T cell, which has a T cell receptor against this fragment of the allergen, then that dendritic cell needs to somehow be activated to put co-stimulatory molecules on its surface. Okay, that means that the dendritic cell has to somehow be exposed to pathogen-associated molecular patterns at the same time as when it was exposed to the allergen. Okay, now this does commonly occur. Okay, generally allergen molecules will tend to be contaminated with pathogen-associated molecular patterns. Okay, so if you think about animal dander, animal skin cells, there is a good chance that when you breathe in animal dander that you're also going to get some pathogens with that as well. Okay, so that could underlie why the dendritic cells, when they've been exposed to the allergen molecule, also get the co-stimulatory molecules on their surface. Okay, but again, it's just hypothesis. Okay, we are still shaky on why this adaptive immune response is initiated in these allergic individuals, but this is what must happen if we are to get the activation of these CD4 positive T cells, which is needed to begin the pathogenesis of asthma. Okay, right, so these dendritic cells do have B7 molecules on their surface, and this binds to the receptor on the surface of the CD4 positive T cell. Okay, now the receptor on the surface of the CD4 positive T cell is a molecule called CD86, okay, which I'll have here. Oh, sorry, what am I doing? CD86. I wrote the right thing, but said the totally wrong thing. CD28 is the receptor for B7. Okay, CD86 is one of the types of a B7 molecule. Okay, right, so here's our B7 molecule on the surface of the dendritic cell, and it's binding to the receptor on the surface of the CD4 positive T cell, which is CD28. This then provides signal 2 to the T cell, okay? And now signal 1 and signal 2 is going to cause a response in that CD4 positive T cell, but it's not fully activated yet, okay? What it's going to cause is it's going to cause the T cell to give itself signal 3. So let me now explain what signal 3 is. Okay, so basically once a CD4 positive T cell has got signal 1 and signal 2, so let's just draw this here. Okay, so here is our CD4 positive T cell. It's now got signal 1, okay, and signal 2. Signal 1 and signal 2 together are now going to cause a change in gene expression within our CD4 positive T cell. Okay, and this change in gene expression is going to cause the cell to suddenly start expressing two molecules that it didn't express before. Okay, and these two molecules that it's suddenly going to start expressing are the interleukin-2 molecule, okay, a secreted cytokine, and then also a component of the receptor for the interleukin-2 molecule, which is called the interleukin-2 receptor alpha component. Okay, so these are the two things that are going to have their expression induced by the CD4 positive T cell receiving signal 1 and signal 2. Okay, so let me just firstly explain the state of the interleukin-2 receptor in these CD4 positive T cells before having received signal 1 and signal 2. Okay, so if I draw the plasma membrane of our CD4 positive T cell here, this is the extracellular fluid, this is the cytoplasm. Basically, the interleukin-2 receptor is a trimer, okay? It's made up of 3 separate subunits, okay? The interleukin-2 receptor alpha, the interleukin-2 receptor beta, and the interleukin-2 receptor gamma, okay? So let me just colour this in. So one of these will be an interleukin-2 receptor alpha protein, which we'll have here in red. Okay. One will be an interleukin-2 receptor beta protein, which we'll have here in green. Okay. And one will be an interleukin-2 receptor gamma protein, which we'll have here in turquoise. Okay, right. Now, that is the functional interleukin-2 receptor. When you have all 3 of them together, this makes the functional interleukin-2 receptor, and an interleukin-2 molecule combined here and cause some signaling cascade inside the cell. Okay, however, CD4 positive T cells do not usually have the full trimer of the interleukin-2 receptor assembled. Okay, usually the bit that is missing is this interleukin-2 receptor alpha here. They express the interleukin-2 receptor beta and the interleukin-2 receptor gamma, but they are lacking the interleukin-2 receptor alpha. That one's missing. So beta and gamma are pre-assembled into dimers, but it can't do anything until you have the interleukin-2 receptor alpha. Okay, so going back to what signal 1 and signal 2 do to the CD4 positive T cell, it's now going to start making interleukin-2, which will be secreted into the extracellular fluid. Okay, so to show this, the interleukin-2 will be secreted into the extracellular fluid. It's also going to start making the interleukin-2 receptor alpha, which will mean that now that can go up to the cell membrane, and you can now form a full functional interleukin-2 receptor on your cell membrane. And then what basically happens is that the interleukin-2 that you have released into the extracellular fluid will now bind to and activate your complete interleukin-2 receptor, which is at the plasma membrane now, and this then gives the cell signal free. Okay, so interleukin-2 binding to the interleukin-2 receptor gives the cell signal free, and that then causes the activation of the CD4 positive T cell. Okay, so if all of that happens, the CD4 positive T cell is activated. Now, what then happens following activation? Well, the first thing that happens following activation is that the CD4 positive T cell is going to start proliferating. Okay, so proliferation follows. So we're going to go from having one CD4 positive T cell here to now having a whole population of identical CD4 positive T cells that all have this same T cell receptor which is directed against a fragment of your allergen. Okay, so you amplify your number of CD4 positive T cells. Then what starts to happen is they start to undergo the process of differentiation next. Okay, in which they change into actual effector T helper cells. Okay, so they're going to become T helper cells now. And there are three main different types of cells that these CD4 positive T cells can differentiate into. They can become T helper 1 cells, okay, they can become T helper 2 cells, or they can become T helper 17 cells. And we know what the main type of T helper 2 cell that you get in asthma is going to be. It's going to be the T helper 2 cell that we saw earlier. Okay, now all of these three different types of T helper cell, they are all capable of helping B cells to activate. Okay, hence why they are called helper T cells. They are capable of helping B cells. All of them are capable of doing that, not only one type. Okay, and the reason that they're all capable of helping B cells is that they are all capable, they all have a certain molecule on their surface. Okay, and the molecule that they all start to express once they're differentiated, but not before they've differentiated. So these cells here, but not these cells here, start to express the molecule CD40 ligand on their surface. Okay, so if I have one of these differentiated T helper cells here, it will have the all-important CD40 ligand molecule on its surface. Okay, and this is what allows these T helper cells that are now mature to actually activate B cells and help in the activation of B cells. Okay, and we'll come back to that when we discuss the additional allergic adaptive immune response that occurs in atopic asthma. Okay, right. Now, that's what all these three different types of T helper cells share, but what's the difference between the three of them? Well, the three different types of T helper cells all secrete very different cytokines. Okay, so they secrete very different molecules that give instructions for different things to happen, basically. Okay, now we've discussed what T helper 2 cells do. They're the main topic of this video. Okay, they secrete interleukin 4, 5, and 13. Okay. And we've seen that interleukin 5 and interleukin 13 are incredibly strongly eosinophilic chemotactic. Okay, so they recruit eosinophils to the site where those T helper 2 cells are. Okay, we've seen also that interleukin 4 is involved in making B cells become plasma cells which produce IgE. Okay, and we'll explore that in much more detail in a moment. Okay. What do T helper 1 and T helper 17 cells do, just so that you've got a complete picture? T helper 1 cells, the major cytokine that T helper 1 cells produce is something called interferon gamma, IFN gamma. Now this gives instructions to macrophages to kill. Okay, basically it activates macrophages and gives them new rights to use brutal force against pathogens, basically. Okay, I always think of some part of Harry Potter where Sirius Black is explaining to Harry that Barty Crouch gave the Death Eaters new powers. He gave them the powers to kill and not to have to bring back prisoners. That's what interferon gamma does to macrophages. It gives them the powers to use brutal force to kill the pathogen, basically. Okay, before that the macrophage is sort of doing it half-heartedly. Okay, T helper 17 cells, meanwhile, their major cytokine that they release is interleukin 17. Okay, hence why they're called T helper 17 cells. Okay, and interleukin 17 is very powerful at recruiting neutrophils to a site where it's high. Okay, so interleukin 5 and 13 is eosinophils, interleukin 17 is neutrophils. Okay, right, but we're not interested in T helper 1 and T helper 17. I'm just giving you that information so that you've got a broad picture. Okay, we are interested in T helper 2 cells. That's the major type that you produce in this asthmatic allergic reaction, again, for reasons that we don't really understand. Okay, this is what happens. Why it happens is another question. Okay, so you produce T helper 2 cells now that have a T cell receptor that's directed against a fragment of our allergen. Okay, and now we're back to our original pathogenesis. These T helper 2 cells are going to go into the blood, and then they're going to move out of the blood at the lamina propria, and they're going to start infiltrating the lamina propria of your airways, and then we know what happens after that. Then it's the story that we spent so long developing. Okay, right, so we'll call it there for this video, and in the next video what we'll do is we'll turn our attention to the additional allergic adaptive immune response that occurs in atopic asthma, okay, which is the activation of B cells directed against epitopes of your allergen.